The virus, considered to be on the verge of life, is completely unable to reproduce on its own. Thus, it needs to infect living cells and utilize their organelles to create new viruses. This specific virus, a human papillomavirus, has just entered a host through a contaminated needle. As the virus wandered throughout the bloodstream of a pubescent female human lifeform, it came across a 2n46 diploid somatic parent cell entering the metaphase of mitosis. During the prophase prior, the cell had dismantled its nuclear membrane, freeing its single nucleus, condensed its chromatin into chromosomes, and completely replicated each chromatid. The cell now had 26 pairs of chromosomes, each with 2 sister chromatids, but the sisters were conjoined by a centromere. The virus watched as the chromosome within the cell began to line up single file along an equatorial plane. 2 centrioles, located at opposite poles of the cell, released spindle fibers to grip tightly to opposite ends of each chromosome. Then the anaphase began. The centromeres split and the sister chromatids were torn apart, becoming 2 separate chromosomes. The cell noticed the virus, watching curiously, and began to explain the next stage, the telophase.
Watch in awe, young one, as I reform the nuclear membrane over the separated chromosome. Yes, it is indeed time for growth and repair.
2 genetically identical nuclei were formed, and the chromosomes began to uncondense back into chromatin. At the same time, the cell used domain expansion, cleavage furrow, separating into 2 daughter cells which move away through cytokinesis, completing the process. The virus amazed that a cell could replicate itself so perfectly with no external assistance, continued to travel throughout the bloodstream, floating aimlessly. It eventually ended up at one of the ovaries, where it began to observe a SUN46 parent ogonium undergoing meiosis 1. Initially, during prophase 1, the virus saw no distinction between meiosis and mitosis, as the chromosomes were formed and replicated, and the nuclear membrane disassembled. However, the virus soon stood in utter confusion as the chromosomes unleashed their cursed techniques, synapsis and crossing over. During synapsis, each pair of homologous chromosome groups together, forming a tetrad of 2 chromosomes and 4 chromatids. Then during crossing over, the chromosomes swapped portions of their genes with each other. When the metaphase 1 began, the virus noted that the tetrads were lined up along the equatorial plane, still in their homologous pairs, rather than single file, in complete independent assortment. Anaphase 1 began. The tetrads were torn apart, and the homologous pairs were segregated from each other. The ogonium spoke to the horrified virus.
Don't worry, I'm the strongest one at reduction division.
As the cell moved on to telophase 1, splitting in 2, prophase 2 took over. No replication took place. The cells simply prepared to begin meiosis 2. The virus noticed that once again, microtubules guided the chromosome to line up on the equatorial plate, metaphase 2, but then the cells stopped. For 4 years and 11 months, the virus silently watched the cells. Of the 2 daughter cells produced, one was significantly larger. It was eventually expelled from the ovary, where it traveled, slowly dying, through a fallopian tube. A handful of sperm cells appeared, and one was able to make contact with the larger cell. Immediately, the cell moved on to anaphase 2. The centromeres split, and the sister chromatids were pulled apart to opposite poles, just as in mitosis. During telophase 2, the virus witnessed a cleavage furrow divide both cells in 2, and the 4 daughter cells moved apart through cytokinesis. They were now each genetically distinct N23 cells, haploids. The virus questioned the largest of the 4 cells.
Are you the egg because you are not a polar body? Or are you not a polar body because you are an egg?
The cell responded.
I am no longer simply an egg cell. I have united with a sperm cell to become a zygote. Throughout the oviducts and the ovaries, I alone am the fertilized one.
The virus posed another question.
If you were infected with HIV, would you still be able to develop into viable human offspring?
Well, If I didn't receive antibodies from the placenta, I might have a little trouble.
But would you lose?
Nah, I'd win.